Hello everyone. Today we are studying biology class 11 chapter number 4 cell division. Chapter number 7 cell division. Okay, we have started with this chapter. Today we are continuing the chapter. Okay. In this chapter we have already studied, we have started with the process of mitosis. In that we have studied the 4 phases of mitosis, that is prophase, metaphase, anaphase and telophase. After telophase, there occurs cytokinesis, the process of cytokinesis. Telophase means what? That was the final stage of karyokinesis, in which 2 daughter nuclei were formed in the end of the cell. Okay, at the end of the telophase, 2 daughter nuclei were formed. So now let us study with cytokinesis. What do you mean by cytokinesis? Cytokinesis means the division of cytoplasm into 2 daughter cells. That process is known as cytokinesis. I repeat, the division of cytoplasm into 2 daughter cells is called cytokinesis. The division starts with the constriction. This constriction gradually deepens and ultimately joins in the center, dividing into 2 daughter cells. This process of division of cytoplasm is perpendicular to the spindle. This mechanism of cytokinesis is characteristic of animal cell. Okay. And in plant cell, what happens? Plant cells are covered by relatively non-flexible cell wall. Due to this, furrow cannot be formed. Instead, cell wall partition starts to appear at the center of the cell and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of 2 adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed with the 2 daughter cells. Okay, in short, we can say in animal cell, furrow appears in the cell membrane during late anaphase or early telophase, and in plant cell, cytokinesis starts with the cell plate, which is middle lamella between cell wall of 2 cells. You can see here clearly in this diagram animal cell versus plant cell. Animal cell, the furrow is getting deepens. Okay, you can see this is the furrow. Okay, this is the furrow. It gets deepens and ultimately it gets, the wall layer gets divided into 2. We are getting 2 daughter cells. And here we cannot see any furrow. Instead of furrow, there is a cell plate, and that cell plate divides the daughter cells into 2 parts. Okay, so you have to remember this, students. In animal cell, there is a furrow formation that gets deepens and ultimately 2 daughter nuclei are formed. And in plant cell, there is a cell plate, and that cell plate divides the cell into 2 daughter cells. Okay. Next is, so here we complete the process of mitosis. At the end of the cytoplasm, we are getting 2 daughter cells. Okay. Next part is significance of mitosis. Mitosis is a equational division. So the chromosome number is maintained constant. It ensures equal distribution of nuclear and cytoplasmic content between the daughter cells, both quantitatively and qualitatively. The hereditary material DNA is also equally distributed. It helps in the growth and development of organisms. Okay, so I repeat the significance point-wise. Mitosis is important for sexual reproduction indirectly. Then it allows the sexually reproducing organisms to grow and develop from a single cell into sexually mature individual. This allows organism to continue to reproduce through the generations. Okay, so mitosis is playing a very significant role, and it is helpful for the distribution of genetic material DNA. It also helps in the growth and development of organism. Then many other significance are there of mitosis. Okay. Old and worn out cells are replaced through mitosis. We can say that some old cells and worn out cells are also replaced by the process of mitosis. It helps in the asexual reproduction of organism and vegetative propagation in plants. The process of mitosis also maintains the nucleocytoplasmic ratio. Okay. Although mitosis is a very reliable process for preserving the genetic makeup of cells or organism, it cannot introduce variations or new combinations of existing genes. I repeat, the process of mitosis also maintains the nucleocytoplasmic ratio. Although mitosis is a very reliable process for preserving the genetic makeup of cells or organism, it cannot introduce variations or new combinations of existing genes. Okay. Then next part of the chapter is meiosis. But before studying meiosis, we need to study about the death of cell. Cell death we will study. I will tell you some points regarding this. You may think of it as a bad for cells in your body to die. In many cases that's true. It's not good for cells to die because of an injury. For example, due to scrape or a harmful chemical which is called necrosis. However, some cells of our body die. Not randomly, but in a carefully controlled way. For example, during the embryonic development, the cells between the embryonic fingers died in a process called apoptosis to give a definite shape to the fingers. This is a common form of a programmed cell death where cell undergoes cellular suicide. When they receive certain signals, apoptosis involves the cell death, but it benefits the organism as a whole. For instance, by letting fingers develop or by eliminating potential cancer cells. I repeat about the cell death. Body is very good at maintaining a constant number of cells, so there has to exist mechanisms for ensuring other cells in the body are removed when appropriate. There are two forms, that is apoptosis and necrosis. Apoptosis means suicide and necrosis means killing. Suicide means programmed cell death and killing means decay and destructions. Automatically there will be decay and destructions of the plants. Apoptosis means programmed cell death. It is a form of cell death in which a suicide program is activated within the cell, leading to the fragmentation of DNA, shrinkage of the cytoplasm, membrane changes and cell death without lysis or damage to neighboring cells. Okay. So, and next part was the necrosis, means what? Decay or destructions of the unwanted cells. Automatically the unwanted cells will be decayed and delayed. Okay. So, by letting fingers develop or by eliminating potential cancer cells. Apoptosis involves the cell death, but it benefits the organism as a whole. So, death of the cell is also a necessary process. It is not, we cannot say that it is a bad process. Sometimes the death of the cell is also a required process to replace the old and worn out cells. Okay, I repeat, you all may think of it as a bad process of the cell in your body due to die. But in some cases that's not true and in some cases that's true. It's not good for cells to die because of an injury. Okay, which is called necrosis. However, some cells of our body die not randomly but in a carefully or controlled way. Okay, for example is embryonic development. And the cells between the embryonic fingers died in the process called apoptosis to give definite shape to the fingers. Okay, this is a common form of a programmed cell death where cells undergo cellular suicide. Okay, and so thank you for today. So here we have completed the part of mitosis. Okay, here we have completed mitosis, means half of our chapter is done. Okay, only meiosis part is remaining, so that we will continue in our next lecture. So thank you for today. We will continue in our next lecture.